Good morning, my dear students. Welcome to my channel Patel Science Academy. Welcome in the 6 number lecture from the chapter Inheritance and Variation. Dear students, as you know, since 5 lecture, I'm teaching you regarding the different topics included in this chapter Inheritance and Variation. So, dear student, this is lecture number 6, and you are watching Patel Science Academy by Shehzad sir. So, dear students, in the previous lecture I have explained about the intragenic gene interaction, that is the interaction that occur between the allele of the same gene, and it's three example I have explained in my previous 4th number and 5th number lecture, that is incomplete dominance, codominance and multiple allele. Incomplete dominance in the 4 o'clock plant, codominance in the cattles, and multiple allele example in the human being for ABO blood group. Dear students, in this lecture the topic that you are going to know about is intergenic gene interaction. Intergenic gene interaction, the another type of the gene interaction. So dear students, you are well familiar with the intergenic gene interaction, which is also called as non-allelic gene interaction, and it is the gene interaction which occurs between the alleles of different genes present on the either same or different chromosomes. So what is the difference in between the intragenic and intergenic? Intergenic, it is the interaction between the allele of the same gene, while the intergenic, it is the interaction between the allele of different gene present on the same or different chromosomes. So dear students, Pleiotrpy as an example of this intergenic gene interaction we'll see in this lecture. And what is Pleiotrpy and what is pleiotropic gene? Let's see. The definition of Pleiotrpy or pleiotropism: when a single gene controls two or more different traits. It means, dear students, when the different trait inheritance, if it is dependent on the single gene, means this is that only gene which controls these characters, these different characters, which may be at least two or more than two. So if these two or more than two different characteristics is being controlled by the single gene only, so that type of gene is called pleiotropic gene. And that process is that gene is called pleiotropic gene, and that phenomenon is called pleiotropy or pleiotropism. So I hope you have understood about this pleiotropy, in which we call a single gene, that single gene which control two or more different trait, that gene is called pleiotropic gene. So we'll see the example of this pleiotropic gene. Now we'll see the example of this pleiotropic gene. Its ratio is always 2:1 instead of 3:1. Actually we should get 4 result, but instead of that 4 result we are getting here 3 results. Why we are getting here 3 results instead of 4 result? Because in this pleiotropy one organism dies because of that condition. So that's why instead of 4 result we get 3 result only. So how this 3 results comes, let's see. So dear students, as you know, pleiotropy, so far we have treated Mendelian inheritance as though each gene affects only one phenotypic characteristic, but in the however most genes have multiple phenotypic effect, a property that is called pleiotropy, as I told you earlier. And it is the actually Greek word, it has come from the Greek word pleion, which means more. So dear students, in human being there is a example of this pleiotropic gene. The example is pleiotropic allele are responsible for the multiple symptoms associated with the certain hereditary disease such as the cystic fibrosis and the sickle cell disease. So in this lecture I'll explain about the sickle cell disease. In the garden pea plant there is also one more example. In the garden pea plant, the gene that determines the flower color also affects the color of the coating on the outer surface of the seeds. So you can understand here, one gene is controlling the color of the flower, and it is also controlling the coat color of the seed surface. So it may be gray or it may be white. So dear students, this is only one gene which is controlling more than one character. That's why it is called pleiotropic gene. As in the human being, sickle cell disease we are we are having here. This sickle cell disease, why this sickle cell disease comes in the human being, we'll see here one by one. So dear students, let's see this sickle cell disease in the human being one by one, and how this 3 instead of 3 is to 1, 2 is to 1 ratio comes there, we'll see in the detail. Most genes that have multiple phenotypic effect, a property is called Pleiotrpy gene. As I told you, means it is a gene which is having multiple effect, or it controls the multiple characters, two or more characters. So for example, the pleiotropic allele are responsible for the multiple symptoms in the of a certain heredity character, such as I told you, cystic fibrosis and sickle cell disease. So we'll see all these disease. Sickle cell anemia. So dear students, sickle cell anemia, in this sickle cell anemia, actually this is a disease, sickle cell disease, which is caused by a gene capital HBS. So normally oval or donut shape of the blood cell should be there in the body of the normal human being, but instead of resembles a sickle cell or crescent shape. So in this RBC of the, in this disease sickle cell anemia, RBC of the human body get converted into sickle shape. So you can see here the sickle shape, conversion of the RBC into sickle shape, and this is the normal shape. So because of the sickle shape, blood doesn't flow properly, and this sickle shaped cell, it scattered or embedded somewhere in the blood vessels, that leads to the problem of the heart diseases, that leads to the death of that patient. So this is about the sickle cell, which is the blood disease, and in which the RBC of the human body blood get convert into sickle shape. So you can see here one more time, this is a sickle shape of the RBC, and this is the normal shape of the RBC. So dear students, this normal shape or sickle shape, this difference actually it indicates the patient sickle cell anemia patient, because of the presence of that HBS gene. The normal healthy gene is HBA, it is dominant over HBS, and the carrier, which is heterozygous, it contains HBA, HBS. So dear students, if somebody is having HBS two times, so that body is sickle cell. If that body is having HBA and HBS, so that body is carrier. And if body is having HBA, so that body is normal. So we'll see it here. So genes which multiplies the phenotypic effect, one gene affect the more than one phenotypic character. For example, sickle cell anemia, we'll see about the sickle cell anemia, in which the blood RBC cells get changed. So this is that cross which is indicating the sickle cell anemia, and let's see the sickle cell anemia in this slides. These are parents, and their phenotypes are: one parent is sickle cell carrier, another parent is also sickle cell carrier. Carrier means those parent which is containing HBA, HBS, this pair. As I told you earlier, if this HBA is 2 times in the body, that body is normal. If one HBA and one HBS is there, so that body is called carrier, means that body will be phenotypically normal but genotypically abnormal, and it can carries that gene into its next generation. And if two capital HBS are there two time, so that body will die because of the sickle cell disease. So how it comes, let's see. So dear students, the carrier with one more time, normal healthy body will have HBA two time. Carrier heterozygous body will have HBS, and sickle cell anemic body will have HBS two time. And the signs of the mild anemia, as their RBC become sickle shaped, that is half moon shaped only under the abnormal low oxygen concentration. So dear students, let's see this about this cross. So these are two parents. Let's see it about this chart. So as I told you, these are two parents. Their phenotypes are, they are both are carrier, means mother is carrier and father is also carrier. So what does it indicating? Carrier means, why we are calling it carrier? Because it is having uh, HBA HBS HBA HBS. So their gamete, these are their genotypes. Their gamete will be, so their gamete will be likewise one HBA, one HBS. These are the two gametes of this parent, and this parent's gametes again will be same, HBA HBS. Remember, HBS two time is responsible for anemic disease and that body will die. HBA two time will be normal body, and one HBA and one HBS will be heterozygous carrier body. So these four gametes will go there and they will fuse with each other. So let's see how does they fuse. How do they fuse? So if this HBA from this parent, it fuses with this HBA from this parent,